I'd like to welcome everyone here in the room at the National Press Club in Washington, D.C., and everyone who's streaming online. This is a presentation and panel discussion on the release today of what we believe is a very important white paper entitled "The Case for Standards in Life Science Research." And before we get into, I'm going to take about 20 minutes to summarize the case before we have a discussion with the panel. I just need to go through several pieces of logistic information here. First of all, as I mentioned, we're streaming this via a webcast at www.gbsi.org/watchthecase, and this is also being recorded. For those who need it, my PowerPoint presentation is going to be available there as well. In addition, we're live tweeting this event. You can follow along at #TheCase, that's one word. We have Wi-Fi for this and for people in the room, and it is called The Case 1. And we're going to save questions until the end of the panel discussion. That is questions from people in the room and questions from the online community. So, and for those of you in the room, please use a microphone because otherwise people who are streaming this won't be able to hear your question. Okay, I think that was the logistics I needed to go through. Who we are the Global Biological Standards Institute. We, as an institute, a nonprofit institute based in Washington, D.C., we seek to expand the development and adoption of standards to support credible, reproducible, and translatable outcomes in life science research. That's our mission. This diagram describes a synergistic relationship between life science stakeholders and the public, increased standards awareness and adoptions, and innovations in biotechnology. GBSI is working to advance all three of these components. The last piece but it's extremely important housekeeping is I would like to introduce 4 panelists who have agreed to participate in our event today. The first is Dr. Glenn Begley, who's the Chief Scientific Officer and Senior Vice President for Research and Development at Tetralogic Pharmaceuticals in Malvern, Pennsylvania. Glenn also serves on the board of directors of the UK-based Oxford Biotherapeutics and is on the scientific advisory board of several biotech companies. From 2002 to 2012, Glenn was the vice president and global head of hematology oncology research at Amgen. And I think it's also important to mention, and I'm sure Glenn will have a lot more to say about this during the panel discussion, that in 2012, last year, Glenn co-authored a highly cited paper in Nature that described how 53 landmark published papers, Amgen scientists could only reproduce the results from 6 of those papers. And if you're doing the math, that's 11%. But I'm sure he'll have a lot more to say about that. To the right of Glenn is Dr. Mary Lou Ganser, who is the president of the Clinical and Laboratory Standards Institute, CLSI. Mary Lou's also involved in CLSI's Global Health Partnership Program. The mission of CLSI is, as a nonprofit standards development organization, is to develop clinical and laboratory test practices and promote their use throughout the world. She's also been involved in the establishment of the International Consortium for Harmonization of Clinical Laboratory Results, which is a global effort related to the improvement of patient care through increased harmonization of clinical and laboratory diagnostic procedures. To the right of Mary Lou is Dr. Yvonne Reed. Dr. Reed has been a scientist at the American Type Culture Collection, ATCC, since 1980. Her very important research is focused on the use of DNA hypervariable probes for the intraspecies identification of human cell lines. The evolution of this work has led to the implementation of routine screening of all human cell lines at ATCC, as well as other cell banks. Recently, she co-chaired the ATCC Standard Development Organization, SDO Committee, on the development of a consensus standard for the authentication of human cell lines through STR profiling. And then last but certainly not least is Dr. William Bentley. Bill is the Robert E. Fischel Distinguished Professor of Engineering and founding chair of the Fischel Department of Bioengineering at the University of Maryland in College Park. And also he's at the Institute of Bioscience and Biotechnology Research. At Maryland since 1989, Dr. Bentley has focused his research on the development of molecular tools that facilitate the expression of biologically active proteins and is the author of over 250 publications. Importantly, Dr. Bentley has served on advisory committees and panels for the NIH, NSF, DOD, DOE, USDA, and several state agencies, and he's also a co-founder of a protein manufacturing company, Chesapeake Pearl. Bill is a fellow of the AAAS and AIMBE and is an elected member of the American Academy of Microbiology. He's also a co-PI of an FDA-funded Center of Excellence for Regulatory Science and Innovation. So we're really happy to have all four panelists with us today. Okay, I'm going to have a technical problem here. Okay, why make a case for standards? Reproducibility obviously is the foundation of life science research, yet irreproducibility is a pervasive systemic problem. And interestingly, many of you have probably seen this Economist cover story from 2 months ago. It appears, and this was an interesting timing coincidence, because when we started this, this case, I don't think there were as many stories in the mainstream press, but it does seem as if more and more articles, both in the mainstream press and in the scientific press, are being written on the problem of irreproducibility. But I'd like to point out that none of them, at least the ones I've read, are speaking to how we can get ahead of this problem. And our case argues that it acknowledges the fact that there is a serious problem with irreproducibility, but it proposes that through a greater implementation of standards, including policies, guidelines, et cetera, we can get ahead of this problem. And that's going to be the recurring theme of my presentation today. Tell you a little bit about the origins of the case. We, GBSI, commissioned an independent outside firm to prepare this report, Health Advances, in collaboration with Feinstein-Keene Healthcare. And I'm pleased to acknowledge and introduce Marie Schiller, a partner, and Olga Coleman, a consultant from Health Advances, as well as Marcia Keene, who's chairman for strategic initiatives from Feinstein-Keene Healthcare. Thank you for joining us today. Let me also tell you a little bit about how the report was prepared. It's based on nearly 60 in-depth interviews, as you can see from the figure on the slide, and this is also figure 6 from the case for those of you who are following. The people that were interviewed were leaders from academic institutions, industry and industry associations, investors, government agencies, foundations, journals, professional societies, standard development organizations, and policy and patient advocacy organizations. I think also it should be pointed out that the results the results also include extensive literature reviews and an analysis of data from sources such as the NIH, the NSF, and pharma. Okay, so let's talk about the case. Clearly, the life science research landscape is changing. It has been changing for the last 10 or 20 years. There's an increasing complexity, and there's certainly an explosive growth of molecular-based techniques and fields. Just think of genomics, for example. There's been a growing emphasis on translational research, which puts basic research that much closer to the bedside as opposed to the bench. There are collaborative approaches being emphasized, such as clinical translational science, CTSAs. There's growing competition, which leads to economic challenges for a variety of reasons, including and especially a growing or a diminishing pool of funds from major funders, starting with the NIH. And the result is all these things sort of contribute to a decreased effectiveness of the way we used to do research, the traditional processes, the peer review and self-correction mode to maintain quality. That just does not appear to be good enough anymore. The other thing that really came out very clearly from our research, our case, is that this irreproducibility issue is a pervasive systemic problem. Interviews conducted for this report with a wide range of stakeholders and also from work that's been published by groups at Bayer and the Amgen group that Glenn Begley headed indicate that, and these numbers vary, but minimally 70% and as much as 90% of published findings cannot be independently confirmed or reproduced. That's a shocking statistic. And obviously, irreproducibility has extensive negative and lasting effects. So it's a systemic problem, but it fundamentally stems from undefined variance in reagents, in practices, in assays, between laboratories, within laboratories. And that's another way of saying that there's an absence of a unifying standards framework for doing research. And I'm just going to point to this figure. This is figure 12 from the case. It's interesting that about 70% of academics, in terms of the responders, clearly do not have any kind of formal quality system in the way they do research. Now, that's inverted if you look at industry, so maybe we'll all go home feeling a little bit more comfortable about how the private sector does research, but I think it's important to note that as pharma itself decreases its own pool of discovery research, they're going to rely more and more on academic research. So if academia, I hate to single out academia, I was an academic for many, many years, but clearly there's a problem within academic research, and again, there's a whole series of factors and pressures that point to that, but it's an interesting inversion as pointed out in figure 12. We believe, as I mentioned in my introductory remarks, that standards can be a solution, if not the solution, for this problem. Standards are an effective and feasible solution to reduce variability and improve research quality and outcomes. All stakeholder groups that were interviewed for the case agree that there is a need for additional standards in life science research. I don't think there's an agreement on how those standards should be implemented, but everybody agrees that there should be standards. But it's also clear that the adoption of standards requires a community consensus, both around the necessity for standards and their content, and that's going to be a challenge. So before I go on, I think it's important to define what standards are. Standards, generically speaking, are an agreed upon way of doing something. Standards can be quantitative or they can be qualitative, and they're in biology, there are two kinds of standards, or in life science research, there are two kinds of standards. There's material standards, and that really refers to highly characterized physical substances, such as biological reagents, cell lines, what have you. They're also called reference materials or reference reagents. And the other major category of standards are written consensus standards. Those primarily are documents that outline community consensus around practices, processes, and procedures. This fleshes that out a little bit more. We affectionately call this our avocado model. And that's just to say that in the dark green on the upper right, the material standards primarily refer to reagents and assays, but there's more of a continuum of standards as they refer to in document standards, and they can encompass protocols, good experimental practices, data analysis reporting, laboratory maintenance, quality systems, etc. Do standards matter? We think they do. And this is the slide that, if you're falling asleep, I'm going to try to wake you up and hope you can follow this. This is our funnel slide, and just bear with me. The left side of this is a hypothetical situation here. The left funnel really represents what's in place right now in the continuum of the research process. And what I mean by that is primarily the academic research process where an idea gets a grant review. There's also a continuum through intra-laboratory principal investigator review. There's peer review for papers that are submitted or publication papers that are submitted to journals for publication. There is colleague replication, and there's self-correction. So if you go down to your local research university and ask a professor or principal investigator if we really need standards, he would or she would probably say, well, we've got this check and balance system in place. And for the most part, as depicted by the gold balls here, which depict quote unquote high quality research as opposed to the blue balls, which depict low quality research, one would argue that this may or may not be an acceptable amount of high quality to low quality ratio here. The argument is that with standards in place, there will be a much, much higher percentage of high quality work that filters through this check and balance system, that the old way is really no longer sufficient. How do we advance biological standards? As I mentioned, it requires the engagement of a diverse group of stakeholders, and I think that without the buy-in of all these stakeholders, this isn't going to happen. And those include, as depicted here, academic institutions, industry, policy and professional organizations, nonprofit funders, journals, regulatory funding and government agencies, and of course, standard development and quality organizations. GBSI would like to provide, we are providing a global independent forum for these diverse stakeholders to ensure that biological standards development is focused on priority needs. Okay, we want to be the link to these different stakeholders that are in place here. And I think that without all of the stakeholders at the table, we're going to get very frustrated very quickly, we, the life science research community. So standards are the solution, and I want to challenge everybody here in the room, online, if you're going to view this as a video, we're actually taking a call to action here. We're calling on the stakeholder groups, these various stakeholder groups, to work with us to establish policies and procedures leading to a more prominent use of standards, which would then result in positive outcomes. To take an example, academic labs and institutions, they're going to need to ensure best practices incorporated into research, into the research process. They need to produce more reproducible results for obvious reasons. They need to protect their own reputations. The same is true for journals. The same is true for industry and investors. And in fact, for industry investors, I think I would be concerned about the best return on my investment in terms of the kinds of reproducible data that's coming to me. So ultimately, expediting the process for the development of standards that can be implemented is going to have a positive effect on research, on development, and ultimately on cures and preventions to enhance human health. This isn't a sort of abstract academic argument here. This is going to have a very, very serious both economic impact as well as an impact on the quality of life as we move forward. So we can do this. This is my last formal slide. This is the only quote I'm going to read. The slides have been have contained quotes that came from the interviews, from the case. This is the one I want to read and leave you with in terms of my formal presentation. The life science research community is creative, inventive, and innovative. It's a community that has found cures for diseases and extended human life. This community can solve the problem of irreproducibility, and a solid standards framework can unify and accelerate this global effort. Thank you.